Welcome everybody. Thank you for taking the time to listen to what all the great features have been put into our new version of DRAGEN. We have a lot of material to cover, so let's jump directly into the slides. So as we do with all our software releases, it's a combination of new updates and new features that we bring onto the platform, and then we do updates to our existing pipelines and software features that we have. So at the first, we will put forth new features that we are introducing for the first time onto our platform. So we have highlighted 4 features that we will cover at the very beginning before we jump into some other details. So the first feature that we are excited to bring to you is the DRAGEN ORA compression. So this is a compression utility that is now onboarded onto the DRAGEN platform, and we will talk into some detail on what the compression utility is and how it can save costs to DRAGEN users. Also, for the first time, we are bringing into a general software platform for DRAGEN biomarkers, which are TMB and MSI. Also, we are introducing an HLA typing pipeline for the first time onto the DRAGEN platform in this release. And another feature that has been requested by our customers for a while, which is the DNA Amplicon feature, is now being introduced onto the platform as well. So in the past, we have had support for enrichment, but in this release, we are bringing in the Amplicon feature for DNA, and then in a future release, we will also be introducing the RNA support for Amplicons. So let's jump onto the first major feature that we are really excited to bring onto the platform is the ORA compression. So what does ORA stand for? ORA is something that we are referring to as the Original Read Archive, and this compression utility gives you lossless, reference-based compression of FASTQ files. So again, this feature is specific to FASTQ.gz compression, and there are 2 steps that we include as part of the compression utility. One is an ultra-fast mapping algorithm that we have introduced into the compression utility. And what the mapping step essentially does, it takes the reads from your FASTQ file and maps against a reference genome that is part of the compression utility. And it determines where does the read perfectly match with the reference genome and where the differences are with the reference genome. And as a second step, we encode that data as is typically done with compression software, and we basically make a record of where those perfect matches and those differences are. We encode the compression field into the records, and that tells us the quality of the compression at that particular field. And of course, we include all the information specific to the file into a header file. And what the DRAGEN ORA compression does is it includes its own internal reference. This internal reference is a human reference, meaning the compression that you will get today with this compression utility is best with human data. It will still work with data from other species. However, the compression ratio that you will get would be lower than what you would get from a human reference. The mapping step that is done as part of the ORA compression is internal to the compression software, as I said. So this is completely different from the mapping that occurs that you're familiar with as part of your secondary analysis. The workflow to get the compression is fairly straightforward. So the data that you get from an Illumina sequencer typically will go through a BCL to FASTQ conversion step to give you a FASTQ.gz file. This FASTQ.gz file is input to the FASTQ.ORA compression utility, which will generate that FASTQ.ORA file. What you get is a compressed file that can be then uploaded to either a local storage or a cloud storage, and then this FASTQ.ORA file becomes an input to the various DRAGEN pipelines that you're familiar with on the platform. So here are the key points that I want to highlight for this compression software. The DRAGEN ORA compression occurs before the mapping step. So the input to the software compression utility is either a FASTQ file or a FASTQ.gz file. For any compression that we do, we save an MD5 file. There is seamless integration with native ingestion of these FASTQ.ORA files into the DRAGEN mapper. So once you have compressed the files, all the DRAGEN pipelines will be able to ingest this ORA file. So you do not have to decompress this file back to .gz to input into the DRAGEN mapper. The DRAGEN ORA compression is enabled on the command line, and it can be run in sequence or in parallel, which means that you have the ability to run with a number of threads that are available onto the DRAGEN platform. And the number of threads can be changed depending on how quickly you want to run the compression software. The default is 8 threads, and you can go all the way up to 16 threads. The FASTQ.ORA files can be decompressed using the DRAGEN 3.8 software, or you could download a standalone decompression software from Illumina's website, and that will let you decompress this file on any node anywhere else you want to do that. So what we are trying to highlight here is that the FASTQ.ORA files are completely portable, which means to decompress these files, you do not need access to a DRAGEN server or any DRAGEN license. The same decompression software has also been made available as part of the NextSeq 2000 in the past. So even if you compress your files using the NextSeq 2000 sequencer, which already includes this functionality, you should be able to decompress and port those files. So again, the compression can be done on the DRAGEN server, but the decompression can be done anywhere, and hence these files become easy to distribute and easy to port. Here we are highlighting the unique combination of high compression ratio, fast runtimes, and low memory usage with this compression utility. First, let's jump into the compression ratios that one would get with FASTQ.ORA files. What we are showing here in this slide on the y-axis is the compression ratio versus FASTQ.gz. The different colors that you see are basically various Illumina instruments and the data that comes out of it, and the compression ratio that you can achieve from there. What we are showing at the very top and giving us the highest compression ratio are our newer instruments. So the NovaSeq 1000 and NextSeq 2000 are the instruments because of their two-color chemistry and how the quality files are binned, quality metrics are binned, you would get the best compression ratio. And you can see that the compression ratio averages to about 5x for the NovaSeq data. That starts to then diminish as you go to our older instruments like the HiSeq 3000 or 4000. So depending on what sequencers you have, and a combination of that, you will see varied compression ratio, but still the compression that you would get would be definitely improved from what you would get from a FASTQ.gz file. The runtimes are extremely fast. So if you use a DRAGEN server to compress the FASTQ.gz file, you will see that the compression for a 30x whole genome is about 8 minutes, and the decompression for that file is about 3 minutes. The compression is about 3 times faster than what you would get with a PIXZ or a multi-threaded gzip version running on the same node. With the same compute resources, the decompression is about 5 times faster than what you would get from PIXZ. One of the bigger issues with compression software is the amount of memory that one requires. With the ORA compression, we also see that the memory usage is extremely low. We are seeing that it requires less than about 4 gigabytes. And again, the compression speed and the time that you see here is purely running the compression utility in software mode. So none of these, the processing that requires the compression has been done on an FPGA yet. So we are purely using the CPU and the multi-threaded capability to do this compression today. There is minimal overhead for ingestion of these compressed files if you are using the DRAGEN pipeline. So here is some benchmarking that we did. So the first plot shows you if you use a DRAGEN server locally and you have a pipeline that does mapping and all the calling on a 100x whole human genome, then the analysis time for a FASTQ.gz file is about 1 hour and 40 minutes. However, if you were to also input a FASTQ.ora file, then the analysis time is the same. So you're basically having a compressed file, but you are not slowing down any of the analysis that you can do. Similar metrics for just doing the mapping, you can see that the analysis time is very close to each other. We also did the same on a AWS F1 instance. And once again, the analysis time to do the whole, to complete the end-to-end pipeline, you can see with a FASTQ.gz or a FASTQ.ora, the analysis time is very close to each other. So once again, you can compress your files and let DRAGEN ingest those compressed files with no loss in analysis time. The advantages of adopting DRAGEN ORA for cloud storage are therefore very evident, right? You can have 80% savings on storage costs. Here we are assuming that you have 5x compression with your NovaSeq data. You have 80% savings on transferring costs. So if you are moving the files from cloud to cloud or between regions within a cloud, or you're moving them locally from a cloud, or going and uploading those files, again, all those transfer costs saved because what you're essentially doing is moving around smaller files. The savings to get this compression are right away, and I will talk a little bit about that in the upcoming slides. And again, because these files are going to be 5 times faster, the transfer times are going to be faster as well. Here are some example benefits of adopting DRAGEN ORA for cloud storage. So if you have 2,000 whole human genomes, we are estimating that you would save about $55,000 on FASTQ storage costs. The assumptions here are we are comparing against FASTQ.gz. The compression ratio is 5, and files are stored for 1 year in hot storage and 2 years in cold storage. Savings on data transfer from AWS to local. So you have about 200 whole human genomes per month is what you would transfer. You are saving about $1,300 per month. If you are saving, you're also saving on data transfer if you're moving files from one region of AWS to another. And again, when you have a smaller FASTQ file, you are saving time in terms of transferring those files back and forth. And once again, if you're using nodes to transfer those files, those will again help you save some money. So again, just all the benefits that you can get, and those benefits quickly add up in terms of dollar signs when you are transferring files that are smaller. If you were to adopt this DRAGEN ORA compression format for on-premise, it means that you would delay the budget for additional servers. What this means is any of the storage costs that you would incur or that you are growing into, those could be delayed because now you are saving smaller files. You could also compress your existing files, and in that way you can reduce your storage footprint. The transfer times are again 5 times faster. So if you're moving files within your network, if you are sharing those files with other users within your network, again, those smaller files are going to be moving around much faster. So when I said previously that the savings with this format will start right away, it means that the DRAGEN ORA license is now included in your existing DRAGEN license at no additional cost. So example is that if you have a level 1 license with 100 terabases, it now includes 100 terabases for analysis and 100 terabases for compression. So connected users will be able to use the compression software as soon as they install DRAGEN version 3.8. And if you're connected, then the license for compression will automatically get downloaded to your DRAGEN server. If you are an offline customer, meaning your server is not connected to the internet, then you can contact our customer service and you can request the DRAGEN license that you would need for compression. So once again, you can download the software today, install it onto your server, and you can start compressing your files. These files could be new files that are coming from the sequencer, or if you have existing files today in your local storage or in your cloud storage, you can download them, compress them, and then put them back. So again, this utility today, in addition to the NextSeq that we had introduced in towards the end of last year, is now available for all DRAGEN users who use the DRAGEN server today. Okay, let's jump to the next new feature, which are the biomarkers that we are introducing into DRAGEN. The first biomarker that we are introducing is TMB, which stands for tumor mutational burden. The DRAGEN TMB offers a customizable and integrated caller for calculating TMB in panels, whole exome, and whole genome tumor samples. We say it is a customizable pipeline because it works with a variety of samples, and also the user can define the regions, and it can also give flexibility to customers to use this pipeline. Hence, it's fully customizable. It is an integrated caller because we integrate the TMB analysis with the rest of the pipeline that does the calling as well as the annotation. One thing to note over here is, because of the customization that we provide in this caller, the user is responsible for evaluating and optimizing the settings based on a specific use case. So once again, we had introduced the TMB feature as part of our TSO 500 panel, but we started to realize that there is a general utility for having such a caller within a general DRAGEN software. So again, we think this is an emerging biomarker and something that our users can benefit from. This TMB module is completely flexible, and it can be computed over custom regions across a variety of sample types. This makes it possible then for users to optimize their use cases and depending on what their needs are for the various samples that they are running. Here we are showing an example DRAGEN command line, and some of the command line options are where the tunability or the customization can happen. So if you see here, there is a VC callability tumor threshold parameter, which is set to 50 in this example, which essentially tells you the coverage that you can set for calling TMB. Also, you can enable the VAF threshold via the command line, and you can introduce a bed file that essentially tells the TMB module the regions that it needs to run over. So again, this customization is completely at disposal to the customer, and they can then adjust these settings depending upon their sample type. Here is the workflow that does the TMB calculation. What we have shown in stars over here are the parameters that the user can adjust. But here are the high-level steps that we go through for running the TMB caller. You start off by doing standard variant calling. And again, the samples could be a tumor only or a tumor normal. And what you get out of that standard pipeline are VCF files for variant coming out of our variant caller. The next step is a region detection step. So here are the coding regions that you would specify with a bed file that you would provide. And you would also specify the minimum required coverage. We then compute the eligible regions, and then we bring in our Nirvana annotation capability that is also inbuilt into DRAGEN to do the filtering based on the bed files and the coverage that you have provided. Finally, the last step is the TMB calculation, where again you provide the VAF threshold, so we will filter based on those VAF thresholds and then make the TMB calls. The output of the TMB caller is essentially a metric file that will tell you the number of calls, it will tell you the eligible coding regions, but it will also give you the non-synonymous TMB and the associated calls with that. So it's a fairly integrated step, but still gives you all the capability to tune it based on the type of panels or the exomes or the whole genomes that you have. The next feature that we want to highlight is MSI or microsatellite instability. Once again, like TMB, this is an emerging biomarker that we had introduced as part of our TSO 500 portfolio, and we wanted to bring it into a more general format into DRAGEN so that users who have their own panels can benefit from this feature. This feature is limited today to a tumor normal matched input. This, like the TMB, is customizable, meaning you could provide a coverage threshold as we show over here in the command line, and then you can bring in a microsatellites file, again over which we would call MSI regions. So once again, this is something that the user can bring in, but the user is also responsible for evaluating and optimizing these settings based on their specific use case. The workflow, again, is fairly straightforward, and in the orange stars is where we indicate what the user settings or what the user needs to provide. So you can start off with a tumor FASTQ or a normal FASTQ, or you could start with a tumor BAM and a normal BAM. And that essentially is input to the MSI caller. There are two user-provided inputs. One is the microsatellites file. So again, these are the regions that you would want to call MSI over, and also the threshold. The output of the MSI caller is a report that lists all the unstable sites within those regions. The MSI caller is a simple module that again takes in your tumor allele counts and your matched normal allele count distribution, again coming from the BAM files, and it runs a chi-square test for sites with sufficient coverage. Again, this is something that you provide as an input. We aggregate across those sites and generate a report, which essentially is a percentage of the unstable sites that I show over here. We also list what were the total number of sites that were assessed, and then a percentage of the unstable sites. We have done some benchmarking for whole genome as well as whole exome samples. These samples were determined or were part of a Promega PCR gold standard array, and then the MS status was determined from those assays. Here on the left in the plots, we are showing how DRAGEN MSI Calling compares for whole exomes and whole genomes when we compare against those gold standard assays. On the y-axis, we are showing an MSI score or a percentage of the unstable sites, and in red is where we are indicating MSI H, which is basically MSI high or unstable sites, and in the greenish or the bluish plot over here, we are showing MSS, which is basically MSI stable or MSI low sites. On the right, we are just giving you an example of how an MSS sample looks and an MSI H sample looks for a tumor normal pair, where you can see for a stable sample, the repeat lengths are well aligned for the tumor and the match normal. However, for MSI H sample, you can see that the frequency of the repeat length is much higher for the tumor pair. Also, just to give everybody some clarity, we are comparing how these biomarkers come about and compare with the TruSight Oncology panel that Illumina also has. So we are just trying to show you the differences in terms of the implementation of these biomarkers for the TSO 500 portfolio and what's available in DRAGEN 3.8. The differences are mainly where the TSO 500 is a tumor-only portfolio, whereas the biomarkers that we have in 3.8 give you more flexibility. However, the thresholds and the parameters for TSO 500 have been optimized and validated to give you the best performance, whereas in 3.8, it's the user that is responsible for setting those parameters. The TSO 500 portfolio is optimized for UMI samples, whereas the DRAGEN offering in 3.8 is not optimized. So again, we are giving you the functionality, we are giving you all the flexibility, but it's up to the user to do all the adjustments and to make sure that the thresholds and the bed files they provide are correct and accurate based on the sample that they are running. The next feature that we want to highlight is the HLA typing pipeline. So the HLA is a set of genes that encode the MHC. It plays a crucial role in the patient's response to the immune checkpoint blockage therapy in cancer. So in 3.8, we are introducing the HLA typing capabilities, and it relies on a nucleotide to protein alignment, and it uses an ILP, which is the integer linear programming method, to find the class 1 HLA allele that can be aligned to most reads. Here is an example command line that we want to highlight, again showing some flexibility and customization that a user can do. You have the capability to bring in an HLA bed file. Again, this basically indicates to the pipeline the HLA regions that you want to run the pipeline over. You also have the flexibility to set zygosity thresholds and tiebreaker thresholds as part of the pipeline. Something, again, the user can customize depending on where they want to call the HLA alleles. Here is the workflow for the HLA pipeline. So you can start off with a FASTQ file or a BAM file. The FASTQ file will, of course, go through the mapping step. If you already have a BAM, then it injects directly into the HLA pipeline. If you are using standard human references, AG19 or G38, we do have the capability to auto-detect the HLA regions. However, you can also bring in an HLA bed file, and that indicates those HLA regions that we want to run the pipeline over. Based on that, we will extract the HLA regions, and then we have a fast HLA DRAGEN aligner that we have implemented that runs over these extracted HLA regions, There is the integer linear programming based typing that we do, as I indicated earlier, and then depending on the allele frequency that you provide as an input, we go through the tiebreaker steps, we determine the zygosity, and the output is HLA alleles out of this pipeline. We have done some accuracy benchmarking. This was done over some whole exome samples as well as some TSO500 samples. These DNA samples were received from the IHWG group, and these are a combinations of TruSeq DNA exome samples. There are some TSO500 samples, and these were HLA typed using the DRAGEN HLA pipeline on a DRAGEN server with an HLA tiebreaker threshold of 0.97. What we also did was, in addition to comparing against the known truth for these samples coming from this group, we also ran concordance against another popular HLA typing tool called OptiType. And what we are highlighting here is that we have high concordance with the data, with the known truth, but also with OptiType. So again, that's something that if you're familiar with the OptiType pipeline and have used it in the past, this is some benchmarking we have done, and we have provided to you the samples and the information related to how we did this benchmarking in this slide over here. So if you want to compare, if you want to run against those and compare 3.8, then you have the ability to do that. The one feature that we want to highlight as part of our HLA performance is the runtime. So running these whole exome samples and the TSO500 samples for those for that data, you can see that DRAGEN runs extremely fast, this HLA pipeline, compared to OptiType. So for some of the whole exome samples, we averaged about 4 minutes of analysis time, while OptiType was about 83 minutes. Again, a big difference in some of the TSO500 samples. So while we have good concordance with the OptiType, you can see that the speed of analysis for this HLA pipeline is extremely fast. Okay, jumping to the fourth major update that we are bringing into the version 3.8 is the DNA Amplicon feature. So this is the first version of our Amplicon pipeline. Again, this is something that has been on our radar for actually a couple of years. We had enrichment always available, but the Amplicon feature was missing. So what we have done is we have optimized the pipeline for DRAGEN that does the mapping and the variant calling to support Amplicon panel. This leverages the existing DNA pipeline, but it does need a few modifications. essentially what we are introducing between the mapping and the variant calling is this feature called primer trimming. And this essentially is the primers that are basically the amplicons, and you can provide that information via a bed file. This is really the on-target alignment records, which will then tag against the amplicon names. And also as part of the pipeline, we do not need to do any duplicate marking. So it's a small change that we have put in, but it's something that is very critical to the Amplicon pipeline that we have introduced between the mapping and the variant calling step, which is essentially this primer trimming option over here. So in addition to this capability that will be available on the DRAGEN server, we will also release a BaseSpace app that will let you run Amplicon samples through it. Here is some benchmarking that we have done. So on BaseSpace today, we have a DNA Amplicon BaseSpace app, and we have compared the accuracy of that app with DRAGEN 3.8. Here we are looking at the SNP and the indel accuracy for recall and precision. And you can see that the recall and precision between the two apps for SNPs is very close. If the y-axis, you can see it's extremely compressed. It's almost 99%. And higher. So this is very close. But however, the indel accuracy is far superior with DRAGEN compared to the existing DNA Amplicon BaseSpace app that exists. Of course, what you will also get with DRAGEN compared to the Amplicon BaseSpace app is the speed that comes with its mapping and the variant calling. Okay, so those were the 4 major features that we wanted to highlight that were new in 3.8. Now let's jump onto the existing pipelines and see what are the updates that we have made there. So going to the small variant calling, if you remember in 3.7, we had introduced this concept of joint detection of overlapping variants in our variant caller. This was a feature that had helped us get extremely high accuracy in the precision FDA challenge that had happened in the summer of last year. In 3.7, we did not enable this feature by default because we were hitting some runtime issues, and we didn't want to make the analysis slower for our customers. So that feature was available, but it was disabled. And customers could enable it by default if they were willing to take a little bit of a runtime hit. In 3.8, what we have done is we have addressed that issue. So now the local runs or runs on the DRAGEN server are about 4% faster on 3.8. with joint detection enabled versus 3.7 without this feature. So we have not only resolved the issue of speed up, but now because it's enabled by default, you should see accuracy improvements with this feature. With the F1.4x, if you run this on the cloud, we still are running a little bit slower. But again, one of the main reasons there is that the F1.4x has a smaller number of CPU cores, so we have not been able to fully optimize the feature over there. But again, what you get out of the joint detection feature is in this next slide, which is the high variant calling accuracy. So if you're running in the cloud and if you're willing to take a little bit of a hit on the runtime, what you will get is an improvement in accuracy. The slide is a little bit busy, but what we are showing on the left is SNP accuracy, on the right is indel accuracy. The accuracy comparison here is essentially the number of errors, which is FP plus FN. And the benchmark is against the version 3.6 caller, which is in blue over here. And if you look at the yellow line, which is the latest 3.8 with joint detection enabled, you can see a big difference across the board in the accuracy for SNPs and indels. So here is what you would get. So if you're running a slightly older version of DRAGEN, if you move to 3.8, then you will see a big improvement in accuracy. We have compared and benchmarked this. On the x-axis, you can see there's a variety of samples that we run as part of our accuracy benchmarking, covering various genome in a bottle samples, various coverages, various instruments, various library preps. So we are seeing, the results to be good across the board with this feature. In 3.7, if you remember, we had introduced this graph genome concept. Again, this was a solution that we had developed as part of a Precision FDA challenge. And that time we had targeted this graph genome to be only specific to AG38. In 3.8, we have now more references that are supported. So if you're using GRCH37, AG19, then you will be able to now use the graph genome with those references. We also had a minor fix that we have made on the NM tags for BAMs that were generated for these graph genomes. And now we have also confirmed that this BAM file that you would get from the graph genome is now ingested by all the downstream pipelines that DRAGEN offers. We have further plans to work on the graph genome. For example, in phase 3 that would come in a subsequent release, we'll be looking at MHC regions, and also how to better handle alternate contigs and population haplotypes that help to improve the quality of these graph genomes. One change that we have made between 3.7 and 3.8, in 3.7 you were able to construct your own graph genome for AG38. Now that we are introducing more references in 3.8, we have removed that capability to build your hash table for a graph genome, but we are making the graph genomes in terms of a DRAGEN hash table available on the Illumina website. The main reason for doing this is to build the graph genome, we need additional files like leftover files, the population haplotype files and others, and that makes the installer or the package to install and download and install DRAGEN very large. So hence we removed that from the installer, but what we have done is we have given you the pre-built hash tables, and that is something that you can go to the product files page on the Illumina DRAGEN support page to download, and then you can utilize it with your pipeline. Here are just some accuracy improvements that you would get from this graph genome. So here we are showing on the left first is the AG38 for the AG002 sample and the FP plus FN reduction for SNPs and indels. So this is something that was already available on 3.7. In 3.8, we have the GRCH37 reference, and you can see that there is improvement in SNP as well as indel. However, the improvement is a little bit lower than what you would see on the AG38, but this is really because some of the difficult regions that are now being addressed by the graph hash table are not included yet in the GRCH37 or the AG19 high confidence bed. So as that high confidence bed improves, like it did for the AG38, then one would start to see the FP plus FN with the graph hash table drop further. But just to point out what the differences are in this slide. Now let's quickly jump onto the somatic small variant calling update. So we are introducing this auto detection of hotspot regions. So once again, this is a feature that we've been meaning to bring in for a while, and now we have curated, I would say, about 5,000 unique hotspot locations that we are bringing in, and this feature will auto detect these hotspots depending on what reference that we are using. So we will cover the standard references, the AG19, GRCH37, and AG38. What it does is it identifies hotspot regions where the risk for genetic mutations is elevated, and it will improve the SNV caller sensitivity with a very small impact on its specificity. This feature is on by default, and you can override or disable this feature if you want. On the right, we're just showing you a quick example of how this feature works. So if you have a sample, and in that sample, if there is a variant, but the number of reads that are supporting this variant is small. However, if there is prior evidence based on a hotspot location that a variant here should be called, we will call that as a variant. If there is no prior evidence and there are fewer supporting reads, then we will not call this as a variant. As I mentioned earlier, we have about 5,000 unique hotspot locations, and we have collected these hotspots based on studies done at the Hartwig Medical Foundation and at PCGR. And here are some references that we are providing from where we have curated these hotspot regions. Here is just an example of what happens for a sample that we had taken. So here is a relevant call at this location where the somatic quality is extremely low and we do not call this variant. However, with the hotspot available, this variant does get called and we now have a boosted SQ or the somatic quality score, and we will also tag that call with this hotspot. So that way it's clear to you that this variant is being called because it's boosted, it exists in the hotspot file, and that way you can filter if you want based on this. Another feature that we are bringing in, and again, this is something that we are pulling in from the capability that exists on some of our other callers, like the small variant caller for germline, the structural variant caller, is this ability to force genotype for small variants for the somatic variant caller. And what this feature essentially does, it lets the users provide key positions that they want to be genotyped and reported in the VCF, even if there is no or low evidence of that variant. Again, the use case are essentially forced genotyping of clinically relevant variants to ensure true negatives. You can also check the VAF of germline variants in the tumor sample, and also multi-sample analysis is one of the use cases that lets you do this. So again, something that we have carrying forward from our learnings from other callers and bringing in this more customizable feature to force genotype into the small variant caller for the somatic caller. Another feature that we have spent some time to assess its performance is the FLT3 ITD calling capability in DRAGEN. So now, based on our analysis and some improvements that we have done, we can see that the DRAGEN caller is now capable using a combination of the small variant caller and the structural variant caller, and depending on the FLT3 ITD event size, that our analysis and our accuracy for calling these events is very good. We have improved the recall for long indels in the somatic small variant caller, and with the combination of the SV caller, we are now able to get high recall and precision for calling these FLT3 ITD events. What this means is, in the past, if you have used a specialized caller, such as Spindle, now you can just use the DRAGEN pipelines to be able to call these events. The plot on the left over here we are showing is we are comparing DRAGEN against Spindle for recall and precision for 25 TCGA samples, with each one of these samples having one validated FLT3 ITD event. The pipeline to run this is standard. We are not introducing anything specific or a new caller. We are still utilizing our standard small variant caller for somatic calling and the SV caller. So depending on the event size that you intend to call, you can use one or both, and then you can take advantage of getting the VCF from both the callers. The small variant caller typically calls events which are about 75 base pairs and lower. The structural variant caller will call events which are 50 base pairs and higher. So depending on the event size that you're looking at, you can either use both or just one or the other, depending on what the size of that event is. The next update I wanted to mention is on the structural variant calling. Like I was mentioning in the somatic variant caller, the forced genotype capability, in the structural variant caller, we had support for SV insertions and deletions with forced genotyping in 3.7. In 3.8, now we are introducing support to forced genotype tandem duplications and break ends as well. So just an extension of the forced genotype capability from 3.7 to 3.8. Also, there is continued improvement in germline accuracy, so the recall for insertions and deletions now exceeds 72% and 61%. while still maintaining a very high precision. If you are comparing against BWA, MEM, and a MANTA, a 1.6 caller, which is the caller that's available on Git today, then you can compare and see that the recall, insertion, and deletion is extremely high. This benchmarking has been done on the NIST genome in a bottle HG002 sample with this truth set information that we provide. So again, our continuous move to improve the structural variant calling accuracy with MANTA that is built inside a DRAGEN compared to what's available in Git. Some quick CNV highlights that we want to share over here. So we have made some updates on the somatic whole genome calling for CNV, and also there are some updates that we have made for exomes and small panels. One thing that we want to highlight as part of our somatic CNV tumor normal analysis is the identification of subclonal CNVs. So in the past, we did have very good support where we would call subclonal CNVs. And what we would essentially do is we have identified that subclonal CNVs end up being called as a ref call. And that's the best assigned state. So we have introduced a feature within our CNV calling, which we call as head-based calling, which I will show in the next slide. And these are what help us to call some of these subclonal CNV events. So as I was saying, that there is a feature that is built into 3.8 where what we refer to as head-based calling. So if you have this feature turned off or you were in a pre-3.8 release, then those subclonal CNVs essentially end up being called as a ref, and you can see that the CN value is 2. With the head calling feature turned on and is default in 3.8, now we are correctly calling the CNV events, in this case, in this example, as a loss or a deletion. We are tagging those calls with this head tag over here, so that's something that you can filter on, and now we are correctly calling that same event with a CN value of 1, which was being missed and being called as a ref in the pre-3.8 release. Again, just a more diagrammatic representation of where some of these events are now being correctly called. So where the CNF value used to be 1.8, we are correctly calling that a CN1. These events would have been otherwise being called as a ref at CN2 are now being correctly called with the right CN values as CN3, CN1, depending on the previous example that I was showing in the previous slide. So again, something that is a little bit more specific, but addresses, if you are looking at the subclonal CNVs, this is something that we have improved upon in this 3.8 release. The other feature, again, that is giving you some additional capability as a user is now you can do segmentation via a bed file. So DRAGEN CNV caller has its segmentation step, but however, if you want to use a bed file to specify segmentation boundaries, now there is capability to do that. So you can use this command line option called CNV segment bed and provide a bed file. And what this bed file will let us do is it will let you specify genes or regions of interest, and those are the regions of interest that we will segment and call upon. And then when you're using this segmentation bed file, then you would use the same bed file for PON generation, so the target intervals are consistent across all the samples, and then the records in the output VCF as part of the CNV analysis will reflect the intervals from those user-defined CNV segment bed files. So again, just some additional capability if you are interested in defining your own regions to segment and call upon. The other CNV updates that we have done are listed over here, and some of them are, there is now a DRAGEN metric for mismatched normal detection. So if you're using the DRAGEN CNV pipeline for whole genomes for tumor normal, there is now a parameter called the outlier BASF fraction. This ranges from 0 to 1. It's a new metric that will tell you how mismatched your normal is versus the tumor. Again, the higher the value, it indicates more mismatch, or there is substantial cross-sample contamination or other contamination that has happened that is making it less matched to what is expected. So just something as a metric that you can use to make sure that the normal that you are using is correctly matched to the tumor. Another key update that we have made is the support for mixed gender panel of normals. So in the past, if you have used whole exome to do CNV calling with DRAGEN, we had recommended that you have a set of normals for the male samples, set of normals for the female samples, and you use them separately. But now you can use the mixed gender panel of normals, and DRAGEN will handle this internally so that we don't have to maintain two PONs. Also, we have expanded the support for de novo calling for CNV. So again, if you're using whole exomes or whole genomes to do CNV calling, then there is expanded support for trios to do de novo calling. A quick update on the CYP2D6 caller. So on the CYP2D6 side, if you remember, we had introduced the CYP2D6 caller in 3.7. The 3.7 caller was based on a Sirius tool that is developed by Illumina's algorithm team, and it was on the 1.0 version of the Sirius caller. The Sirius caller has been updated to 1.1, so we are now bringing in that capability into DRAGEN. And there is a big improvement in accuracy. So if you can look at 3.75 and 3.8, you can see that the accuracy now is about 99.5% based on the 212 samples we have for the CYP2D6 calling with validated truth. So again, a bunch of improvements that have happened in the Sirius tool that now we have integrated into DRAGEN that bumps up the accuracy to 99.5% with 3.8. So something that again is extremely large update for us in terms of this 3.8. Quick set of updates on the RNA-Seq. We now have an rRNA, or what I would say ribosomal RNA filter that we have now included as part of the RNA mapping capability in DRAGEN. Again, this is essentially to address the large fraction of reads in RNA-Seq experiments, which are the ribosomal RNAs. This feature essentially has an rRNA decoy which will map your reads against that, and then those rRNA reads are filtered, and you'll get a report of the percentage of the number of rRNA reads that you had. The main purpose of this is that you will get faster runtimes, you will get smaller output files, and again, improve performance and stability of downstream tools that really do not need to worry about these rRNA reads that you might have. So this is extremely important because in some of the experiments that we have done, you can see that the percentage of the rRNA goes all the way to about 20%. So now we have a capability built into DRAGEN to help you filter those out. And here we are showing some benchmarking that for some of these samples which had these very high percentage of rRNA, the analysis time now has dropped compared to what we had in 3.7. So again, just some enhancements to the pipeline, but definitely the benefits of doing this are extremely large, where you can now get smaller files, cleaner files, and then better support for any other downstream tools that you might be running which do not need those reads. We also have made an important update to the single-cell RNA pipeline that we first introduced in 3.7. So in this version of 3.8, we are introducing the genotype-based demultiplexing feature in our single-cell RNA pipeline. So what this lets you do is it enables analysis of multiple samples in one single-cell RNA library prep without the unique barcodes, and it will also let you do doublet detection. So this essentially helps you reduce the cost because now you are multiplexing multiple samples within a single library prep. Here we are just showing you a quick example of what the demultiplexing step does. It needs an input, which is a VCF file, which includes the genotype information for the samples that you have in your experiment for single cell. And using those genotype information, we are able to then identify the various samples that you had multiplexed. The benefit, as I was saying, is that you would have lower costs in your experiment. In the past, users have used a tool called Microsatellite to have this feature. We have also done concordance against this Microsatellite tool, and here are some benchmarking that we have done internally. So we have taken two data sets. The first data set had about 360 million reads. As the experiment, there were 32 samples that were multiplexed in that experiment. We have very high concordance with Microsatellite. However, the DRAGEN runtime to doing this demultiplexing step is much smaller. You can see it takes about 3 minutes for DRAGEN. It takes about 22 minutes for Microsatellite in this experiment. In the same time, we are able to run the whole single cell RNA-Seq run. So while this difference is not too much, while you go to a bigger experiment where you have much larger number of reads, you can see that the DRAGEN runtime is still fairly short for doing the demultiplexing, about 8 minutes. However, the Microsatellite runtime is very high. And in the time that it takes to demultiplex, we are able to finish running the whole DRAGEN single cell RNA-Seq run. So again, something, if you're using a single cell pipeline, something that you could try with DRAGEN, not only is it running faster, but now you have the ability to save some costs if you're interested in running the demultiplexing option with DRAGEN. Wanted to jump to some major update that we have also done on our methylation pipeline. So if you running any methylation experiments, then some of the tools that are widely used are called Bismarck, BWMeth. We also have a DRAGEN methylation pipeline that has been there as part of our offering for a long time. But the traditional way to do alignments with these tools is that depending on the protocol that you are using for methylation, so directional, non-directional, there are multiple passes that you have to do through the aligner for each one of those. So it could sometimes be 2 times, it could be sometimes 4 times, depending on your protocol. And as a result of that, what you are getting is longer analysis time. In DRAGEN 3.8, we now have a new mapping approach for methylation. So rather than aligning reads to the candidate strands in separate runs, so as I was saying, there are multiple passes through the mapper, we now have modified the aligner hardware within the DRAGEN FPGA to now do per read alignments simultaneously in a single run. And what that essentially means is much lower analysis times if you're using DRAGEN's methylation pipeline. So here we are doing some comparison. Again, Bismarck, BWMeth running on standard CPU cores, extremely slow to run these methylation experiments. DRAGEN, with its multi-pass approach, that is pre 3.8, was extremely fast, but still was taking a couple of hours to run some of these methylation samples. Now with a single pass, you can see that there is a big improvement in runtime, even just comparing the DRAGEN's previous version. So this is extremely important. If you're running methylation experiments, you're going to save a lot of time with the single pass option. And this option is now enabled by default in DRAGEN. So again, a huge time saving if you're running a methylation sample. A quick update on the expansion hunter or the repeat expansion tool that we have in DRAGEN. What we have done, there are 2 updates that we're bringing in. One is that we were not doing a very good job in 3.7 for the androgen receptor repeat genotyping. So as you can see that as the size of the AR repeats had hit about 40, then we started to flatten out over there. And this is extremely important because the pathogenic nature of this starts to show beyond 40 repeats. So here, if you had any of those samples, you would not be able to clearly detect if it's pathogenic or not because we're not able to show those repeats. In 3.8, we have improved this functionality, and now you can see that for these samples, we are clearly showing that, there are repeats that go beyond 40, and now you are able to better diagnose if, there is any pathogenic, content or not. Also, what we have done as part of the update for Expansion Hunter is introducing additional genes. So again, something of interest if you are interested in the repeats related to these genes, then they have been added to the catalog, and also some which are specific to AG38. Another update that we have made into the 3.8 software is the read trimming capability in DRAGEN now. So if you remember, in 3.7 we had introduced this poly-G trimmer in specific to poly-G trimming. In 3.8 we are now introducing an hardware accelerated generalized trimmer. So what this lets you do is it lets you do trimming that is similar to a functionality of an Atropos, a cut adapt, or a Trimomatic. However, it is multiple times faster. It is integrated in the DRAGEN workflow. It supports hard as well as soft trimming. So you can set the trimming to say remove the reads that you are trimming, or you can still keep them in your BAM files that are generated at the end of the mapping, but they will not be used. So again, a lot of flexibility into this trimming capability that we have built in. And again, this is part of the mapping step. So you are not incurring any additional steps. You are not bringing in a new tool. You can enable this trimming as you are doing the mapping and the aligning in DRAGEN. Also, if you want to just trim your FASTQ files without mapping, that capability exists. And the various options that you have make it extremely customizable and flexible for you to use. So in addition to the poly-G part that I mentioned, now you can trim based on loquals, on adapter sequences. So even if you want to trim, if you haven't done it as part of the BCL to FASTQ, you can now have the capability to do it as part of a DRAGEN mapping step. Or if there are any ambiguous bases that you are aware of. So it's a more generalized trimmer. So something that otherwise if you would have to go outside of DRAGEN, do the trimming step, bring the FASTQ files back in. You don't have to do any of that. It's all built in, and you can run it as part of your standard workflow from your FASTQ files that are coming out of your BCL to FASTQ conversion. A quick update on the Nirvana annotation. So in 3.8, we have updated the Nirvana version in DRAGEN, but the most important update is now it's integrated with the DRAGEN run. So in prior versions, somebody had to first generate a VCF file, and once the VCF file is generated, then you had to run the Nirvana tool standalone to generate the annotated file. Now in 3.8, you have an end-to-end capability where you can start from your FASTQ, and the output in a single step will give you a Nirvana annotated JSON file based on the VCF that DRAGEN generates. Just some quick updates. If you're a BaseSpace user for DRAGEN, we're going to introduce 2 new DRAGEN apps. So there's a single cell RNA app, which will now be released as part of 3.8. So this is a new app that will come on the platform. And there will, of course, be the new DRAGEN Amplicon app that I mentioned. We will update the existing apps that are there to bring them up to 3.8. And then we are improving for the RNA and the methylation apps, the metrics and the reporting that go with it. Again, this is something that has been fed back to us from users, so we are excited to listen to them and also make those improvements to provide the right amount of metrics and the better reporting that they were requesting from these apps. Just a quick shout out to our ICA team. So this is the new Connected Analytics Cloud Platform that we are introducing, that was launched at the beginning of the year, and there are demo pipelines for DRAGEN that are available as part of ICA. So all of those pipelines will now be updated to the 3.8 phase. And just a general announcement, we have been getting a lot of questions around support for CentOS 8. And what are we going to do now that CentOS 8 is no longer going to be supported in general at the end of the year. So that was something that we had planned for this year, is to have DRAGEN support CentOS 8. But now we know that that is not going to be supported in general. So we will continue to release CentOS 7 like we do today. But as Illumina, we are assessing what would be the next Linux OS that we would get to. And again, this is not just something specific to DRAGEN. Our instruments also use Linux. So this would be a more Illumina-wide approach to align on an operating system that we think has the necessary features that we need, but also the legs to carry us for multiple years. So we're still assessing what would be the next update for us from CentOS 7. But in the meanwhile, we will continue to support CentOS 7. I think that goes all the way to 2023 or 2024. And then we're looking at what we need to do for the next update of the OS. Yeah, so I think that's the last slide. We have some appendix slides that would be made available to you as well, which gives you some additional information. So please feel free to look those up, when you get those slides dropped into your mailbox.